So, I don't see Ranjit here. Is he here? Oh, there. Oh, okay. I was looking all around. Okay. So, the next talk is by Ranjit Padinhateri from IIT Bombay. Excuse my pronunciation of the name. My name is no easier to pronounce, actually. So, Ranjit is going to talk to us about the nature of intrachromatin interactions in the three-dimensional folding of chromatin.
Can you hear me at the back? Yeah, thank you, and thank you for the organizers, Devay and Sandeep, for inviting me. So I'm going to talk about three-dimensional folding of chromatin, and some of you, so I was just telling that I have given this talk in Max Science before, so all the Max Science people might have heard at least first part of the talk, but there is something at the end which is new, which is a little bit newer than what I gave last time. So let me just quickly go through what three-dimensional folding of chromatin is. So the word condensate or condensation has been used in the context of chromatin for the last 40 years or so. So chromatin condensation is a known phenomenon where the DNA, which is the bare DNA, condenses into a structure which we call chromosomes or chromatin, right? So DNA is not bare in the cell, it is condensed by large number of proteins into forms of chromatin as shown in this electron microscopy picture here and kind of a Gen AI picture there. Now the question is, what is the nature of condensation? And it turns out that whatever the specific nature or the way the DNA condenses decides the fate of the cell. For example, if the cell becomes a neuronal cell or the cell becomes a skin cell is decided by the way the DNA locally condenses. So therefore it is important to understand how it is condenses in space and time. So it has a very specific spatial organization the way it condenses, and it has also a temporal organization, and all of this matters for all the processes like gene regulation, recombination, translocation, repair, spreading histone modification, and so many different processes. The 3D organization and the local condensation matters. And one of the experimental method to understand the precise nature of how it condenses or locally folds is a method called chromatin conformation capture experiments. And let me quickly tell you what this is. So basically DNA is some polymer, and as it folds, different parts of the polymers will come together and interact, and this method allows you to figure out these contact points. So what you have is that you add a cross-linking agent that cross-links wherever they are folded and in contact, and then you can add a enzyme that cut the DNA at random locations, and that would leave and separate, that would make a pieces of DNA like X. For example, it cuts here, 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 and here. It would generate a piece like X, which is a piece of DNA, where this is one part of the DNA and this is other part of the DNA. And then you can separate it out, and then you can sequence. Then you can sequence that X-like part. So this part might come from one region of the DNA, and while the other part might come from far away region of the genome. And these two X and Y can be in contact, and these experiments can measure what is the probability that X and Y was in contact, and for any X and any Y, that is like a matrix. So you can see that experiments can generate a contact matrix which says that what is the probability that a segment X was in contact with segment Y. So this is the Y and this is X, right? So this is for chromosome X, some particular region 90 megabase to 100 some megabase, both same, and this is the contact matrix, and this squares here says that they are locally condensed. So you can see that they are locally condensed here, 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 and these two regions again interact, forming this kind of interactions. So there is, this already tells you that there is some condensation plus some few condensates interact together. That's what this broadly tells you. So once experiments can generate this contact structure. Now the question is, given this kind of a contact map, how do you go back and generate a 3D structure? Because experiments can only provide a two-dimensional structure, contact map like this, and from this you have to generate a three-dimensional structure. And it needs some polymer physics, and we have been thinking about how to go from here to here, and this is something that we have been working for last few years. One of the important points to remember while going from here to here is that the contact probability between segments I and segment J is always very small, typically like less than 10%. So this means that it is highly dynamic structure. Things are like in some senses liquid-like if you wish, and things are changing with time, and therefore it is not trivial to go from this picture to that picture. Yeah.
So in the NMR spectroscopic structure determination of proteins,
Yeah.
there's a very similar issue that has been solved, right? Which is that you measure the NOEs. You measure the NOEs and then you use the strength of the coupling to determine distances.
Yeah.
And then that's on a two-dimensional map.
Yeah.
And then you use those as distance constraints in modeling the three-dimensional structure.
Yeah.
So why is this going to be so different?
So it is different for a few reasons. First of all, this is an active problem where there is enzymes doing stuff all the time. And the interactions are much more complex. Like there's not like 20 amino acids. Here there's like so many different types of interactions, and the interacting partners itself are binding and dissociating. Their concentrations are changing in time, and they are all active. So there is, if you look at the entire picture, is much more complex. But I fully agree that the idea of protein and how the protein fold can be very useful to think about it, and we will use some of those ideas to think about it as well. Like you will see that some of those ideas that we used was also used for protein folding. So it is useful, but if you want to solve the full problem, this is much more complex. So yeah, so you need a polymer description of chromatin, and we understand the polymer description of DNA reasonably well. DNA has a worm-like chain model, and we know the persistence length and so on and so forth, but we don't know much about the polymer property of the chromatin at all. So, and if you want to simulate chromatin, if you want to start somewhere, you have to start with the polymer, and what would you put the polymer property? If you want to generate, if you want to simulate it on a computer, what will you put as the polymer parameters? And actually there is people heuristically simulate chromatin, but there is no real idea on what those numbers, actual relevant numbers are, and something that we try to work out in detail. So just to quickly take you through this, chromatin is a complex polymer. There is there is a DNA which is folded with the help of a protein called histone to form this kind of a structure, and then those histones further interact and form a higher order structure, right? So now there are many types of interaction. As you can see, there are different colors of proteins, and we don't know what their interaction strengths are, how strongly they interact. All of this information we have no idea at all. And now if you want to, not only that, if you want to model chromatin, you cannot model at the nucleosome resolution. So the natural way of modeling chromatin is, you can say that, okay, this is my polymer and nucleosome is like a bead, and if I do that, one human chromosome is like 1 million nucleosomes. So you cannot simulate 1 million beads. So you cannot even simulate a single chromosome, and we have 46 chromosomes. So that's like 46 million of the order of that kind of beads, and it is impossible to simulate the whole genome as at the scale of nucleosomes. So one has to go into a much more coarse-grained scale where multiple nucleosomes are like considered as a single bead. So any realistic simulation would typically require a bead spring polymer chain where one bead represents a chunk of chromatin, and that chunk could be 1 kb if you want to study a gene region, and that chunk could be like 100 kb or 1 megabase if you want to study the whole genome. So depending on what problem you want to solve, you would use a coarse grain model for chromatin, and the coarse graining level will depend on what exactly the question that you want to answer. But how do you coarse grain this kind of a heterogeneous polymer? That's a question. And typically, let's say models assume a few kb is equal to 1 bead. So, but if you assume, for example, this is 6 nucleosomes. So if you assume 6 nucleosomes as one bead, we don't know what is the size of this bead. Is it like 10 nanometer? Is it like 20 nanometer? Depending on how these 6 nucleosomes are folded. If the 6 nucleosomes are like this, then the bead will have a different size. If those 6 nucleosomes are folded like this bead will have a different size. So even the physical dimension of the bead is not easy, and it's not easy to understand. And this is a question that what is the physical dimension of the bead? Yeah. Yeah. So, I don't know anything anybody has done successfully. Something we tried, not kind of a systematic coarse graining. That's what I'm going to talk about. Yes. So we want to systematically coarse grain then. So if you want to coarse grain, and once you coarse grain, then if you have a coarse grain model, in the coarse grain model we want to know what is the intrachromatin interaction. For example, one region of the chromatin would interact with other region. What is the strength of this interaction? Not only the strength, what is the nature of the interaction? What potential will you put in there? Would you put a Lennard-Jones potential, or would you put some other potential? And what is the strength? What are the parameters of those potential? So there are two questions about the intrachromatin interaction. And not only the intrachromatin interaction, what would you put the spring constant as? Like, is the spring very flexible, or is it kind of a rigid, stiff spring? We don't even know that, because we don't know. Nobody has pulled chromatin at the scale of a gene. All those people who have pulled chromatin have taken the whole chromatin and pulled at the scale of a mitotic chromosome or something, but nobody has pulled at the scale of a gene by taking an in vivo chromatin. It's not at all very easy to do it. Okay, and there are many other parameters. For example, the bendability of chromatin. Is chromatin a semi-flexible polymer, or is it a kind of a more flexible polymer? If it is a semi-flexible polymer, what is the bending stiffness of the chromatin? Does it have an intrinsic curvature? So none of those parameters related to chromatin is not known. And something that I'm going to quickly tell you today is how we determine, or we have some estimate of these parameters. And in particular, I want to focus on the nature of intrachromatin interaction, because that drives a lot of condensate formation and so on and so forth. Okay, now what we know, so I said a lot of things we don't know. What we know, the last 30 years of literature, is something at the nucleosome resolution. Thanks to many crystal structures and so on and so forth, we know that nucleosome has a dimension of about 10, 11 nanometer, and we know that how often the nucleosomes are placed on an average. So we know the typical nucleosome distance and so on and so forth. So since we know if we have a chromatin model at the resolution of a nucleosome, we know a lot of length scales. So what we did was that we started with a contact map data that gave us contact map at the nucleosome resolution. So there is a very specific experimental data called micro-C experimental data, which uses micrococcal nuclease. That's why it's the name micro-C, and it generates contact map at the resolution of a nucleosome. So this is, for example, the data from C et al. Here for example, this dot represents a nucleosome at 31.22 megabase is in contact with another nucleosome at 31.24 megabase. That's what this means. So every dot in this is a contact between two nucleosomes on an average typically. And what we did, so Sangram, who was in my lab, as a PhD student, we made a model to go from this kind of a picture to a polymer picture. Actually, we have two models, and I wouldn't describe the details of this model now. I will be happy to discuss it after the talk, because for the wanting of time. But one of the model is the following, that we generate an ensemble of static chromatin conformation, and the average of those ensemble of conformations should give you this contact map. Now we have a particular procedure, and it has some details, but the crux of that is that you can put highly strongly interactions like springs, which would bring together faraway beads. For example, this dot would mean I would put a spring between this and bring them together. But the spring is inserted with a probability Pij. It's not that always the spring is there. The spring is there or not there stochastically with the probability Pij. So this brings in the stochasticity into this, and the Pij putting in would essentially at the end of it generate this kind of a contact map. So we go around this, follow some procedure, which is a little bit non-trivial procedure, and then generate a large number of static conformations, and then equilibrate those conformations, and the average of those large number of ensemble of conformation would give rise to this kind of a contact map. For example, this is the experimental contact map, and this, if you do the procedure, you will get a similar contact map. For example, there is a square here, there is a square here, and for example here there is a structure, there is a structure here. So we can generate large number of configurations, and the average of that will give back this kind of a contact map.
Yeah, so from this kind of data, micro C, have people looked at like the enhancer promoter sequences and those loops, like the boxes that you see, right? I mean, this blocky structure. Has it anything to do with the actual enhancer promoter sequence? How much has it been probed?
So some of a little bit has been looked at. We have some new work on enhancer promoter with this micro C data, and Anders Hansen is the pioneer in this, who does a lot of work on this. So other than that, I don't know who has looked at it. Okay, so we can generate this contact map, and what can we predict? So we basically, so far we put in what we, and we get what we put in, but we can now predict the 3D distance between any 2 points. For example, if I take this nucleosome and this nucleosome, what is the 3-dimensional distance in nanometers? So we should be able to predict that, and we do that. And here is a prediction. So this is the genomic distance between any 2 points in kilobases, and this is the 3-dimensional distance between those 2 points after folding in nanometers. So for example, if you take two genes which are 160 kb away, and their 3-dimensional distance would be about 280 nanometer approximately. So that's what this says, and this matches with experimental data, which, for example, this is an alpha globin gene. We take the contact map of that particular gene and generate the 3D distance, and it matches. And if you take some other gene, it will not match, and that's what the blue shows for some other gene, because we have to take the right contact map to get the right folding, and then only this will match. So we can predict the 3D distance. Once we have now the 3D configuration at nucleosome resolution, we can coarse grain. What does it mean? We can take, for example, 10 nucleosome and call it a big bead. So we take this 10 nucleosome and draw a spear around it, and we can ask what is the size of this spear. And if you draw many spears along the genome, we can ask what is the distance between those spears, which is LCG. And then if you have multiple spears, if you have 3 spears like this, we can draw an angle, and we can ask what is the angle between them. Now this LCG, the fluctuation in LCG will tell me what is the spring constant, and the fluctuation in theta will tell me what is the bendability. So we can, from this data which is generating large number of conformations and looking at the distributions of LCG and distributions of theta CG, in a coarse grain picture we can get some idea about the stretchability, the flexibility of the spring constant of the chromatin, as well as the bendability, the bending stiffness or bending fluctuations of chromatin locally at every point, which means along the genome we can get all this data, or we can generate this data from micro C map. So that's what we did. So this is along the genome, the size of the spear, radius of gyration, radius of the spear, and of course it increases and decreases. Some places the radius is very high, which means that the chromatin is not folded much, it is kind of open, and some places there is a minimum, which means that location locally the genome is very highly folded. So we can get the radius of gyration along the genome for every 5 nucleosome, 10 nucleosome, 25 nucleosome, 50 nucleosome. So you tell me what scale you want. I want every 50 nucleosome, which is 10 kilobases. I have this curve of how does the radius of gyration vary along the genome. Now we can take an average of this, and that's what is plotted here, and number of nucleosomes versus average RG, and that's what this curve, and this is kind of a control, so to say, is a self-avoiding walk, which we will get n power 0.6, and real chromatin would be below that, and this is the data for real chromatin for different genes. So these are many different genes, and the bluish represents the heterochromatic and reddish represents the euchromatic regions, and there is a difference between those regions. And the dots are microscopy experimental data from Javet Zhuang's lab, and without any extra fitting parameter. The only thing is the size of the nucleosome, etc. we already know. It just falls very close to the experimental data, suggesting that the numbers that we predict are quite sensible. So this is something that we predict again, the size of the chromosome, depending on how many nucleosomes you want to coarse grain. Now that we have it, we can do many other things, and I wouldn't go into the details of this. And yeah.
From the previous slide.
Yeah.
So this deviation that you have from the control,
Yeah.
is that basically because in the way that the chromatin is organized in the chromosome,
Yeah.
there is a return to the same points many times,
Yeah.
because the chromosome also has a sort of large structure within which the nucleosomal DNA is folded and then folded again and then brought together. So the distances can drop for both very large, the physical distances can drop for very large along genome distances, much more than for very short distances. Do you understand what I'm saying? That means that as you go away, you expect that the physical distance should largely increase with the along genome distance.
Yeah.
Right?
Yeah.
But then at some point,
Yeah.
again there'll be a reduction because the thing will fold upon itself
Yeah. Yeah.
because of the compaction. So
You're right. Whenever there is a loop,
behavior,
whenever there is a loop, if you look at single cell specific structures, we will see those. These are average structures. But the reason why it is much below the green is because, as you rightly said, it is folded. So the folding alone will make it much below this. And what you said is right. When there are loops, some regions will be closer than a nearby region, and such information is already buried in this, which I am not showing now, but it will be there.
So in very large considerations, there should also be some periodicity that is observed.
But if you average over many structures, that will go away. But if you look at one specific structure, you can see some of those periodicities. Yeah. Sorry. So this is many of these things as a function of genomic location, and you can see that it has some at the TAD boundaries they have a different behavior, and interior of the TAD they have a different behavior. So this has a connection with the TAD structure. Now we can get, as you said, the distance between two nearby beads and their fluctuation, and that will give us the spring constant. So we can predict the spring constant, and so the typical number for the spring constant that you would want to put in dimensionless unit is like 1, of the order of 1 is the number I would put, or it's about 1 to 10 piconewton per micrometer. That's the kind of number that you would want to put depending on what you want to study, right? Yeah. Oh, long ago here, yeah.
Yeah. So does this at the large number of nucleosome bits, does it go to the fractal globule?
It should show, because we have only gone up to 160 nucleosomes or so.
Right.
Because that's the only thing we could simulate. We have gone up to 400 here. We have simulated 1000. We took the central part to avoid all those end effects. So that what is showing here is the scaling for 400 in the middle. So that when we average, we get all the. So there only this much one would see. But if you do like many thousand nucleosomes, one would show those kind of behavior. Yes. Yeah. So we can then get the spring constant, and these distributions, if you switch off all the interactions, analytic, we can show that it matches with the analytic relation known in when there is no interaction. Now the question is, if you have two chromatin, how do they interact? They interact, so two beads of a coarse grain chromatin, if you put an LJ, Lennard-Jones like interaction, they will not overlap, they would be like this. But in reality, these two polymer segments can mix. Therefore, the interaction between two polymer segments has to be soft, and it cannot be a Lennard-Jones interaction. So this is another point that we make, is that the interaction between any two chromatin segment in a coarse grain model has to be soft interaction, which means that segments can mix, and this we quantify now how much they can mix, and I will come to that in a minute. So before coming to the quantification of that, we can also measure the angles, just like structural biology, protein biology would do. All kinds of angle one can measure, the bending angle and the dihedral angles. So I wouldn't go into the detail, we did all that. So we have this, the bending angle distribution, and I just want to say one thing, that the distributions are quite wide, which means that the inverse width of this is of the order of kBt, therefore it is quite flexible. And then we did an inverse Boltzmann calculation to understand what is the bending potential, what is the interaction potential, and it turns out that the interaction potential is much softer than Lj. So the black is the Lennard-Jones potential, and the interaction potential between two beads of chromatin is much softer than a Lennard-Jones potential. So it has the interaction strength and the softness. So this is the idea, and then if you want to know the strength of the interaction, we can actually predict it from the contact probability, and this is very much like some of those protein ideas, and they also get something similar, that you can predict the interaction strength from the contact map by this kind of a relation. And we have, the good part is that we have this denominator slightly better fixed than many of those models. This is a parameter, and then we can predict the Pij and the interaction strength, and then one can simulate this, and then we can get the 3D distance versus contact probability scaling relation, and it scales in a particular way, like minus 1/4 or 1/3.5, and then we can even predict the 3D distance for any two regions. So this is the 3D distance that we predict for a human chromosome, and this is the 3D distance measured from a microscopy experiments, and they are exactly the same. Note that the scale is exactly the same, is about 100 nanometer to 700 nanometer, and the color precisely represents those 3D distance. So we can accurately predict the 3D distance for a large segments like of 2 megabases, and using this coarse grain model. I'll just take 2 more minutes, and just to say that 3D organization is happening one part by condensate formation, and then another part by a phenomenon called loop extrusion, where some enzymes actively extrude DNA. So they take this DNA and literally extrude loops out of it, and a competition between a loop extrusion and this condensation, or both, is basically leading to the chromatin organization. So we have a model to also incorporate the loop extrusion into the system. So the details are there in this paper, and once we do this, so we ask many questions. For example, what is the role of binding and dissociation of this protein called cohesin to obtain the 3D organization? How long would it take for this gene to organize a position in 3D, and so on and so forth? So we can ask the time question here. If you tell the rate of extrusion and the binding and dissociation of this extruders as parameters, we can know something about the time. And sorry, what am I doing? One important point is that this is not my, it says end the talk. Okay, it's here. So one important point is that if you don't have a intrachromatin interaction, you will not get the kind of contact map that you would want. So even with extrusion, you require some amount of intrachromatin interaction. That's one of the takeaway lesson. And another takeaway lesson is that when there is extrusion and intrachromatin interaction, there can be some regions of chromatin where they compete, and then it is possible that at some locations where domain-specific intrachromatin interaction is there, this can happen in Drosophila and in some part of human, there the condensate can dominate even though there is extrusion. So effective extrusion will not be even seen, because the strong intrachromatin interaction will hold the structure, and the extrusion will have no visible effect if you look at the 3D distance, the contact map. But if you look at the dynamics, if you look at, for example, the relaxation time versus genomic separation, then the extrusion would matter, and depending on the way you extrude, you can get different results even if there is a condensate. So we claim that dynamics could distinguish extrusion versus intrachromatin interaction, while structure alone may not. Average static structure may not. So with this I will stop. To summarize, we studied the polymer property of chromatin, bead spring chain models to describe chromatin, and we predict bead size and all the parameters, soft potential, and some aspects of loop extrusion versus interchromatin interaction. I will be happy to discuss this more, and I will be happy to discuss this more after the talk and during the lunch break, and I will be happy to take questions if there is any. First row only.
Yeah, so there are models,
Yeah.
polymer models of chromosome where they tend to take not a similar type of bit but multi-bit. That means multicolored bit,
Yes.
like 5-color bit.
Yeah.
And where each color can have the interactions which is kind of more preferred than the interbit kind of
Yeah.
interactions.
Yeah. Yeah.
So how different are your model from this, first of all? And second thing is, other thing is that it is shown that if you do not take the chip-seq data or micro-C data,
Yeah.
then what happened that you tend to see the formation of what is called knot in the polymer if you simulate. But once you put high C data or chip-seq data, then you see that those knots are not there in the chromatin structure. Have you checked the probability of knot formation in your model?
Yeah, we have checked the, I'll answer the second first and then later. So we have checked the knots, and we know the way we fold is such a way that there is no knots. But the knot question is not at all trivial. There is a lot of things with soft interaction. In principle, there can be some knots, but even in mitotic chromosome, for example, people have seen knots. So there is recently, like Job Decker measured that in mitotic chromosome, at least the level, the scale they could measure, there are knots, and so on and so forth. So depending on, so knot question is still to be answered. It's a very interesting question that what happens and how, and presumably topoisomerase will dissolve many of those knots. So that's the knot part.
Also, high C data is very population average.
Yes.
It is smear out knots unless they occur with 100% penetrance in every chromosome.
Yeah, yeah,
If they occur like 25% of the time, you not see.
yeah. And about the other model, of course, if you, depending on the detail that you want, so in our model, the interaction between I and J could depend on I and J, for example, epsilon ij. So if that is an effective way of taking, saying that you have a bead, red bead, making ith bead and jth bead interact, that's a kind of an effective interaction model. And if you want some extra details of these proteins diffusing and that will do something specific, you may want to have a model like that. So depending on what question you want to answer, I think you need to take that kind of model. But if you just want to predict 3D distance, this is good enough.
Okay, so Ranjit, it's very nice talk. Last paper that you showed, of course,
Yeah.
I'm aware of the previous stuff, but the last one where you talk about this the interplay between these. So, do you actually consider condensation explicitly, or you're just saying that okay, the beads are kind of interacting?
Yeah, we just take intrachromatin interaction as a kind of a attractive potential.
And you were saying that underlying mechanism can be condensation or
Yeah.
something. That's the,
Yeah.
because there is a paper from Frank,
Yeah.
it's on archive,
Yeah.
where they actually look at this particular question, where you have co-condensation as and looping. They
Okay.
look at the interplay.
Oh, okay, I didn't see that.
So that might be relevant.
Oh, I'll
These condensates may not be dynamic. They will be very fixed. No? You don't have a question? No, see that. Okay. Sita, you have a question? Okay, so thank you, Ranjit.
Thank you.
And our next speaker is Ithai Mani from IIT Madras. Welcome. And he will speak to us about liquid-liquid phase separation driven fibrillization of amyloid beta protein.
Okay, very good afternoon to everyone. I thank the organizers for this opportunity. So I would like to give an overview of some of the work that we have done on this amyloid beta aggregation and phase separation. So the liquid-liquid phase separation is not a new thing. It's been reported, studied, explained a long time ago in the context of globular proteins and colloidal systems. What you see here is a classical, a lysozyme protein, which is a type of a globular protein. And people have experimentally measured what happens when you create a protein solution of different concentration at different temperature. You kind of create this kind of a liquid-liquid phase separation. It basically means that two liquids phase separate, and if you measure the concentration of protein in these two, they basically have different concentration of the protein. So this is just an example, but there are a lot of other examples one can cite. And initial interest on this system were to understand what are the requirements to see liquid-liquid phase separation, more from a colloidal standpoint. So people are actually interested in looking at some models, interparticle interaction models, that could predict this kind of liquid-liquid phase separation, or LLPS in short. What you see on the left-hand side is phase diagram predicted for a Lennon-Jones potential. What you see here is the Lennon-Jones potential between a pair of colloids. And so here we have temperature in reduced units, and here we have density in reduced units. And you see the classical, vapor-liquid equilibrium, and there is a solid-liquid equilibrium, and then there is a vapor-solid equilibrium. Okay, it's very typical of any molecular fluids. On the other hand, if you look at this system, so here this Leonard-Jones potential was taken, but then it was shrunk in terms of the range of the attraction. So the difference between this and this is that you can see the range of attraction kind of cuts off around about 1.4 or 1.3, but here the interaction goes all the way up to some close to 2 times the diameter of the particle itself. So if you make the attraction between a pair of colloids short-ranged, then one can actually predict this kind of a phase separation. You can see that there is a fluid-solid coexistence above, but then this what we know as vapor-liquid equilibrium basically gets buried inside the solid-fluid phase boundary. So this is something one can calculate with this kind of a model. And then further on, people actually made a generalization saying that if the ranges of attraction is less than 25% of the particle size, then you might see an LLPS. Okay, so that was a kind of a generalization. So it can be protein, it can be particles, it can be any rigid object with no internal degrees of freedom. People actually show that this is one possibility. And subsequently, there are also other interest in looking at non-isotropic interactions. Okay, the previous model was an isotropic interaction. So what I mean by isotropic interaction is that the nature of interaction depends only on the separation distance and not on the orientation. Okay, so that's isotropic spherically. But then there's other development that happened where particles were actually shown to have some sort of attractive patches. With regard to protein, for example, we know that there are hydrophobic patches, there are some sort of charges on certain surfaces. So when you look at the interparticle interactions, one can think of beyond the isotropic interaction. So that's where the patchy interaction was brought in. And people showed that even in this kind of a patchy interaction, one can also predict this kind of phase separation which is buried below the fluid-solid interactions. So these are the things that have been already done for quite some time. And one more thing we can also say is that from the colloids point of system, there are other things like there is this a gel through percolation, and there is a glass formation which is also kind of intersects or interferes with the liquid-liquid phase separation. So there's a lot of work being done on this particular field. I'll come back to this slide a little later. For example, here what you see on the left hand side is an experimental work on lysozyme salt conditions. So again, you see this kind of a binodal, the liquid-liquid phase separation on the top, but then when you come deep, you see there is a glass line that actually connects or intercuts the LLPS itself, or the liquid-liquid phase separation. In terms of cartoon, it is shown nicely here that you have this, the red line that you see, that is the liquid-liquid phase separation boundary, but then before it terminates, you see there is a glass line that kind of kind of cuts. So suppose if you bring a system from here to here, you are more likely to get arrested gel or arrested glass even before the phase separation happens. So the point I'm trying to make here is this LLPS is not only just an equilibrium phase separation. If you quench deep enough, you can even interfere with the glass line and percolation line and so on. So the dynamics of liquid-liquid phase separation also is very important to take into account. Okay, on one hand, we can predict LLPS from an equilibrium point of view, but from an experimental point of view, there are a lot of kinetic effects that brings in additional features, additional challenges. So this is an example where the attractive glass lies close to or within the unstable region. So what I mean here is that this black line that you see, that is an attractive glass regime, and that happens to be below the LLPS. This again is from the context of colloids or globular proteins. And I can show more such examples with more intricacies, where in this case, for example, we have a dynamical arrest happening deep inside the LLPS boundary, or you can have this kind of, like I said before, you can have a glass boundary intersecting the binodal. A lot of experimental data that you can see on globular proteins. Now, even though this LLPS was very well known, but I think the recent, recent interest or renewed interest on LLPS happens because of this, relevance to biological systems. Many of you might have actually seen this kind of picture. This basically shows a kind of a cartoon of a cell in which there are many objects which are actually membraneless. Okay, so there are many condensates or droplets which does not confine through a membrane. Okay, so generally we think cell means there is a membrane bounding it, but there are many objects that are found. You see there's a P body, U body, there are so many nomenclature that is out there. All this spherical thing that you see, these are all membraneless condensate, and that's when this nomenclature biocondensate or membraneless organelle came in. So ever since this topic was brought in, then there's a lot of interest on studying liquid-liquid phase separation in macro biomolecules like proteins, RNA, DNA. I think we already saw many such examples in this conference. So now I wanted to kind of now switch to what our interests are. So we were actually interested in studying this amyloid beta, which is a kind of an intrinsically disordered protein, and I will show you towards the end of the lecture that this is kind of a protein also shows liquid-liquid phase separation due to some of its intrinsic dynamical characteristics. So to kind of tell you about what this amyloid beta means, there is a amyloid precursor protein APP which has about around 770 number of monomeric units, amino acids residues, and that's supposed to form some functional role in our body. But this protein also, it's actually a membrane bound protein. Okay, so what you see here is a membrane, and this big stick that you see here, that's N terminal, C terminal, this entire protein is APP. And these proteins are actually subject to enzymatic cleavages through various enzymes. And again, these cleavages are required for some normal biological functions, but once in a while there is a mistake that happens in the cleavage point, and that leads to a formation of an amyloid beta monomer, and that seems to have some harmful effect. For example, in this case, if you consider on the left hand side, this is supposed to be the right enzymatic cleavage. So you have this APP here, one particular enzyme basically cleaves at this point, and this is released, and there is another enzyme that cleaves at this point, and this gets released. So this is a normal bio process. On the other hand, sometimes if the cleavage is incorrect, for example, instead of cutting here, if it cuts around this point, then it basically leaves this the yellow, the green ones, and those are kind of called amyloid beta monomer, and they tend to aggregate. So purely from a biophysical point of view, this is just a mistake of an enzyme cleavage process. So when this, if you look at these segments, typically you might have 40 residues or 42 residues. Sometimes you get 36, 38 also. Okay, so there's a range of sizes one can create. So we are saying that from a 770 sized protein, we make smaller segments which are predominantly hydrophobic, but they also have some charge residues. Later on I will show you that. And we are interested in studying what happens to these amyloid beta proteins or monomers. So what happens is that once those are cleaved incorrectly, and those proteins tend to aggregate, okay, they form aggregates, and then once they get aggregated, also form some sort of a plaque or network that kind of deposits in important tissues like brain tissue, neurons, and so on. And it kind of affects the neuro signal transmissions and transductions. This is a cartoon picture. This is supposed to be like a normal neuron in the brain, and then when you have this kind of an aggregate getting deposited in this tissue, it kind of interferes how these neurons will communicate. So that leads to a lot of cognitive disabilities, Alzheimer, Parkinson, many other neurodegenerative diseases. So from a medical point of view, people actually look at this problem from a PET scan kind of methodology. So what you see here is a normal brain, and this is a Alzheimer diseased patient's brain. And what you see is a lot of this red spot that basically says there's a presence of many of these aggregates. Okay, so they put a dye, and the dye actually specifically binds to these plaques, and that's when you see this red signal here. Okay, and so this Alzheimer is something that one can check through a PET scan and many other imaging techniques. So these are the biological significance of these proteins. Now there are a lot of studies people have done on looking at how these amyloid beta aggregates in vitro conditions, meaning you take this in a glass tube or a test tube and then study how they aggregate and how can we understand the kinetics and what determines those kinetics and so on. One of the very simplest thing you can do for people like me who are not really an experimentalist but you want to get into an experiment, what you do is you do something called this fluorescence assay. It is basically you put a dye, this is called a THT, it's a kind of a dye that has a specific binding depending upon whether the protein is aggregated or not. It would not bind if the protein is monomeric state, it would bind if it forms some sort of an aggregate. Okay. So basically this dye is also fluorescent, so the fluorescent signal depends on whether it is bound or unbound. Okay. So you take this as a more like an assay, a fluorescence assay, and then we can see we look at the fluorescence coming out of this dye molecule as a function of time when we put this dye molecule in the presence of a protein, and if the protein tend to aggregate, you will see an enhanced signal like this. Okay, so here you have a monomeric state, the signal is weak, but then if this aggregates form, if this monomer forms some aggregates and then form fibrils, and then this dye would actually more specifically bind to this, and you get an enhanced fluorescent signal, and you can see this sort of a sigmoidal kind of a curve. And you can detect the kinetics by looking at this simple approach. Now we were actually looking at this problem from multiple aspects. We want to understand what is the mechanism of this amyloid beta aggregation, and how do we interfere or how do we prevent this aggregation through some sort of additives, and then also how do we actually disintegrate or disassemble these aggregates even after they form, whether this is reversible or not. And we are also interested to know whether these aggregates are actually affected by the flow conditions, the shearing effects and other fluid mechanical aspects. And finally, we were also interested in looking at how do this, there are a lot of biopolymers in our body, and that can also interfere with the amyloid beta aggregation. So what does that affect? So these are the set of questions that we have been looking at in the last 5-6 years or so. Maybe I will quickly run through some of the previous work, and then I'll go to the LLPS part. So what you see here is this is a TEM micrograph of those fibrils. You see this thread-like objects, those are the amyloid beta fibrils, and these are the based on the fluorescence assay, you can kinetically study, you can study the kinetics as a function of time, how much fibrils is formed. So what you see, this is the control, that means only protein is allowed to aggregate, and you see all the monomer goes to a complete aggregated state. Now if you want to prevent this aggregation, what you could do is you can actually treat them with some nanoparticles, chosen with respect to the size and charge and so on so. And depending upon how much nanoparticle you put, you can actually arrest or prevent the formation of aggregates. Okay, so we know some mechanism by which we can interfere the course of kinetics. On the other hand, if you start with a fully formed fibril like this, and you want to say how do we disassemble this? So in this case, these fibrils are quite stable, and they will not disassemble by itself due to some small temperature or pH changes. What we need to do is we put some plasmonic nanoparticles like gold nanorods, and then we shine with some NIR light, and what it creates is some local hotspot, and this allows this protein to disassemble and disaggregate. And you see that as a function of time, these protein basically disassembles and disassociates. So these two results basically say that on one hand, if you start with the monomer, and if you want to prevent aggregation, you can treat them with some medicines or like nanoparticle or other things to prevent the aggregation. That's one learning. On the other hand, suppose if these aggregates are already formed and you want to look at a way that you can get the monomers back, so you have to treat with the different kinds of particle and give some thermal energy, and you can also kind of bring it back. So those are some of the things that we learned from this work. And we also looked at the effect of shearing and the effect of mixing, and how these processes affect the aggregation. This is because these monomers can be carried over through our blood vessels, and blood as it flows also subjected to a lot of shear. In the conventional context, people have studied that if you shear an aggregating system, a shear would actually prevent aggregation. Okay, so that's what people have shown before. But in this protein, we found an opposite effect, that if you do a shear effect, it actually kind of does a different role. So here on the first hand, we have a simple co-ed cell, and we apply a uniform shear. In this case, you can see that the control, if you don't do anything, if you just put a quiescent liquid, over a period of 40 hours, there is no much growth. But then if you kind of apply shear, then you actually see that the rate of formation of aggregate gets faster. Okay, so here we sorry?
So this is known and reported. It depends on the mechanism. So when the shear causes the developing aggregate to break up, then you have many more seeds. And so you have much faster kinetics.
Secondary nucleation, So what I was trying to say is that many of the globular protein aggregation, when people look at study shear effects, it used to be a breaking event, mostly breaking event. But here on the other hand, we have this kind of a secondary nucleation phenomena happening to facilitate faster nucleation. On the other hand, if you do a simple irregular mixing, which kind of gives more collision probability between the monomers, then you see a faster rate of formation. So we understand the aggregation kinetics from all these aspects. Now coming to this effect of biopolymers on aggregation, we were interested in on this polyphosphate. Polyphosphate is actually implied on many important biophysical systems, like it is involved in cell signaling, in sort of cell death, inflammation, and even metabolism of energy and so on. So it's a very kind of a common workhorse in our body. Polyphosphate does many such work, and we were interested to see how these polymers might interfere with the polyphosphate, again from an in vitro point of view. So here we did this experiment with two different conditions. This is a neutral pH, and the other one is an acidic pH. So when we say acidic pH, we could be like 4.5 or maybe 3, which is very highly acidic, and the outcomes are very dependent on the pH. Okay, so when you are looking at neutral pH, when we add the polyphosphate of different amounts, it basically suppresses the aggregation rate, just like how the nanoparticles were acting on. Okay, so it has a more like a nanoparticle like an effect to prevent the aggregation rate. On the other hand, when we look at this acidic pH, in this case, the polyphosphates actually seem to enhance the rate of aggregations. So the black one that you see here, that's a control. As you increase the concentration of polyphosphate, you see that the aggregation completed much faster. Okay, so we see that the aggregation rate increases with the polyphosphate under neutral pH. Basically means that polyphosphate promotes aggregation of amyloid beta. This is very significant because when we look at cancer or many other cell inflammation, people have found that the pH in those cells are much acidic. Okay, and the biophysical process happening in an acidic environment can be quite different. So we see that in a normal condition, the polyphosphates are not really enhancing the aggregation, but rather it actually suppresses. But then suppose if there is any pH fluctuation in the cell leading to an acidic behavior, you can actually then do the opposite effect, it seems. Okay, so that's the main takeaway from this aggregation studies. And now we actually go further to see what is the nature of aggregate formation in these systems. Now if you look at the the polyphosphate effect, I hope you can see here, this particular diagram is basically optical microscopic images of the system. We have treated at different, this is at acidic pH. So you can look at that, we have added 40 micromolar, 80 or 200 micromolar for a fixed concentration of the protein, and you see the development of droplets as a function of time. So this droplet formation is a significance, I mean it just basically signifies the liquid-liquid phase separation in the system. And what you can also do is that in the system you can also study turbidity, and the development of turbidity also tells you that there is some growth of droplets. Yeah.
Without the polyphosphate there won't be.
Yeah, no. So if you see here, this pH 4.5 at control, you see that it forms aggregate, but you won't see a droplet. Okay, so it will be just an empty image. I mean, you can't see anything. But if you do TEM, transmission micron electroscope, you will see a fibril structure that I showed in the beginning. You can see that just like how the THT fluorescence assay increased, the same way the turbidity also increases, indicating the formation of this droplets and two-phase initiation of two-phase separation. And again, we are showing that the effect of increase in the concentration of polyphosphates. So this is 40 micromolar, this is 80 micromolar, this is 200 micromolar, increasing in the concentration. On the other hand, when I put 200 micromolar of polyphosphate at neutral pH, there is no turbidity. It's a clear solution. No droplets, no aggregates phase separating. And then we can also do further analysis by looking at confocal microscopy. We can dye the system and we can see the droplets forming here. And we also did fluorescence quenching experiment, fluorescence bleaching experiment to see whether the droplet that we see are actually liquid or not. Okay, so if it is a liquid, if you bleach a spot, it will regain the fluorescence again. It's a very typical experiment to be done to characterize a liquid-like nature of the droplet. So we take a spot, we first bleach it, and then we wait for it, and then it regains the fluorescence. It kind of indicates there is a very diffusive transport indicating liquid-like behavior. And then we can also construct this kind of phase diagram between the concentration of polyphosphate versus the concentration of protein, and you see that one region where it does not form any LLPS. You need a critical concentration of polyphosphate to induce this. The one on the right, the green region here, that's where you see phase separation, LLPS, whereas at low concentration you don't see. So there is always, for a given amount of protein, you always need a critical amount of polyphase to induce the phase separation here.
Is this A beta 40 or
42 also we have done, but I have not shown it here. It does the same way. In fact, 42 is more prone to aggregate. So yeah. And then we can do further. Okay. Yeah.
I have just one question.
Sure.
So, is it just polyphosphate or any anionic chain can do it, because there are like sulfated sugars like GAGs?
Yeah.
So, if they also can exert same effect, or?
Yeah, so I will explain to you later that this effect is coming because of electrostatic interactions between the polyphosphate and protein. As long as the biopolymer that you add has a negative charge, then it should work. Okay, so yeah. So there's nothing specific about polyphosphate other than the charge here. The biopolymer you mentioned, is it charged?
Yeah, so like sulfated sugars.
Sulfated sugars, yeah.
Like glycosaminoglycans.
Okay. We have not tried that, but the principle we are looking at is typically electrostatic driven. So as long as the biopolymer is negatively charged, it should work. Yeah. Yeah. Okay. So so then we further looked into the aggregate formation. So if you see at pH 7, you see that you get this kind of a fibrillar network. This is a normal case. But then if you increase the concentration of polyphosphate, you don't get any fibrils anymore. You get a little amorphous aggregate, which is different from the fibrillar network that you see here. On the other hand, when we look at this acidic pH leading to LLPS, and then if you look at these droplets, if you dry the droplet and if you look at inside, we do see fibrils inside those droplets. So what we are saying is there's a two-step process. In the acidic condition, when we treat polyphosphate and protein, the first stage is to form the liquid-liquid droplet, and then within the droplet, the protein concentration is enriched, and that leads to an aggregation as if it would happen in a bulk system. So you have a liquid-liquid phase separation followed by liquid-solid phase separation, or crystal growth in other words. So that's what you see, that inside these droplets you see a large number of fibrils that are formed. So we say that the fibril formation is actually amplified, enhanced due to the formation of liquid-liquid phase separation. Yeah, people have done it, but we have not done it. People have done it. Actually, in fact, if you have LLPS, compared to the average background concentration, the concentration inside the droplet can be 100x in other experiments people have measured. We have not measured though.
So I have a question. The morphology of the fluid will still remain the same if it is sort of in a droplet versus bulk?
Yeah.
Is
So
it the same morphology?
we see a little rough fibers in the LLPS compared to the bulk experiment, but looking at fiber level structural information is a little tricky. You have to go beyond TM. You have to do some people have done AFM kind of a study to see the local pitch is different, local structure is different. But from this same analysis, we found that these are a little thicker and rougher compared to the bulk fibrils. All right, okay. So basically, I think the key message I want to show here is this effect of pH and polyphosphate in the aggregation behavior of amyloid. I started 5 minutes a little late. I hope I'm not. Okay, okay. Just 2-3 more slides. Yeah, I know that all of you are hungry. Always, the talk just before the lunch is always the worst time to give a talk. So, anyways, I'm hungry as much as you are. So, if you look at the amyloid beta, cartoonistically we have shown like this, a monomer system, and this is a polyphosphate. When you have amyloid beta polyphosphate at neutral pH 7, the overall charge in the amyloid beta is still negative, but there is a region in the protein that is prone to aggregate, okay, and those regions have actually positive charges. There are a couple of residues which have positive charges, and due to the electrostatic interaction between the negatively charged polyphosphate and the positively charged local aggregating region, that basically leads to prevention of aggregation, prevention of amyloid fibrillation. So that is the hypothesis we are making here. On the other hand, when you look at acidic, the entire amyloid beta, there is a lot of positive charge groups, and then again the polyphosphate still remains negative regardless of the pH. In this case, we are talking about like an oppositely charged mixture of polymers. That we know that if you have oppositely charged mixture of polymers, they would phase separate through electrostatic coacervation, and that's what you see, that the leading to the formation of droplets, and within the droplets, due to the enhanced concentration of the fibril proteins, you end up getting fibrils. So that's the kind of mechanism we have, and the way to test this, I'll probably skip this secondary analysis. And the way we can test this analysis is that we can do a molecular dynamic simulations. So again, this is a 40 residue long time scale experiment. So what we did is we took a small region where they will aggregate, and then we treated with the polyphosphate, and we looked at the how much amount of beta sheets and alpha helix and other structures are there between the two pH. This is initial results. We are not seeing the complete story here, and there seems to be some suggestion that from the simulation that there is more beta sheet formation in the pH 3 compared to pH 7, even at a single monomer and polyphosphate level. But then going forward, we want to look at a more coastline model to see a phase separation in the full scale system. This also I will skip. The one last point I want to make here is that so far all of us have been working on DNA, amyloid, and RNA, those kind of biologically, human-related proteins. But there is a whole lot of proteins out there which are intrinsically disordered, but are there in the plant world. So what I am showing here is a seed, a cross section of a corn seed. And in the corn seed, you see there is this the dotted region here. Those are the starch and gluten. And if you do a SEM analysis, you will see that there are a lot of small droplets that you see here that are shown by the arrow. And these are nothing but protein droplets. And this has a lot of resemblance to the membraneless organelles that we showed in the in the animal cell before. So we are actually interested to look at how these protein droplets are forming in the seed, and this is something one can see in variety of seed proteins. Okay, there's also a nice book on seed protein to talk, that kind of tells you what is the composition of seeds. So I want to draw your attention that while one can do DNA, RNA, and proteins relevant to our health, human health, but there is also equivalent set of problems to be studied in the plant world also, and we hope that we can make a lot of useful study in those domain as well. So with that, I thank you all, and I'm really happy to take your questions.
Thank you, Etharaja.
Thanks.
Do people call you Mani or Etharaja?
Idaya is fine. Idaya. Yeah. My name is as complicated as you were saying about yours.
Yeah, absolutely. Yeah.
going to an international conference is a mess, I fear, because every time they pronounce it so differently. Even in North,
Just say money.
yeah,
Money, everybody, even if they cannot money, they
Yeah.
can say money.
yeah.
Money is fine for
So when I was in college, and the instructor was handing out answer sheets,
Answer sheet,
corrected
yeah.
answer sheets, a bunch of them.